Today, we are going to continue on the computational challenges in cancer immunology and also cancer immunotherapy. In the last lecture, we talked about several aspects of cancer immunology and cancer immunotherapy, which includes what mutations are in the tumor, what mutated proteins or peptides are being presented on the cancer cell surface, and whether there are immune cells infiltrating the tumor, and how much each cell types are in the tumor microenvironment. And we started describing T cells in the tumors and the importance of understanding T cell diversity and T cell clonal expansion in the tumors and in the blood. So in the early days, people did TCR sequencing. They also have done T cell single cell RNA-seq together with T cell sequencing. And I also want to mention another algorithm which was developed in our own lab for getting the TCR sequencing directly from tumor RNA-seq data. So we mentioned that in the tumor profiles in the early days, pathologists want to get tumors that are really pure. But the reality is that in that tumor, there are always immune cells infiltrating, including T cells and B cells. And the infiltrating T cells express T cell receptor at the RNA and protein level. And so if we actually look at the reads that are mapped to the T cell receptor sequences, for example, this is in TCGA, you can see some RNA-seq reads that are mapped to the V segment of the T cell receptor gene segment in here. If you remember in TCR, this is actually a BCR situation. You would have V and D and J, and then when these are stitched together, you get the closest CDR3 is what makes a T cell receptor or B cell receptor. And so if we just map the raw reads onto the gene, this whole genetic regions, you can see that on the V region, there are reads that are actually mapped there. This is because the V segment is kind of long. And if your read is shorter, you can map that whole read onto the V segment. The tricky situation is when the read is mapped to the D segment and J segment, which are very short, especially if you map to the junctions and creates insertions and deletions. So in those regions, very often the read is considered unmappable to the genome and thrown out as garbage. It turns out the V segment and the J segment has some amino acid patterns. And so if you have an unmappable read, but it has this type of amino acid sequence patterns on the V or J, we could consider it as a potential candidate read, even though in the middle or one end of the read is unmappable to the genome, because this unmappable region could be the junction sequence, which is hypervariable and not in the reference genome. And so even if one end or partial read can map to the V or J segment, which contain the VJ amino acid pattern, we will rescue them. And then with all these reads that are kind of only partially mappable to either the V or J segment, we can then do de novo assembly to really create the contig regions and then really assemble the CDR3 sequence. And so using this approach, we can get both T cells and B cell receptor sequences from the unmappable regions of the RNA-seq. And so we have done this for all of TCGA. This is one early study that we published in 2016. We look at each TCGA samples and we calculate the TCR and abundance of each TCR in each patient sample. And so remember, if you just do TCR sequencing from the tumors, you will get, this clone happened 20 times, this one 5 times, this one 3 times, and so on. For TCGA, you cannot really get additional tumor samples to do the TCR sequencing anymore. For many tumor samples, the tissue biopsy also is very valuable. If you already used it, it's a very small amount of tissue. If you already use it for DNA sequencing and RNA sequencing, there's often not enough material to do additional TCR sequencing. So if we could get the TCR and BCR directly from the RNA-seq, that saves us a lot of effort. And also, TCR sequencing is quite expensive. It costs about $600, and BCR sequencing costs close to $1,000 a sample, whereas RNA sequencing takes only $200 to $300 a sample. And so when we analyze the TCGA data, for each TCR, we calculated its diversity using Shannon entropy, or you can use some other approach. For example, we use something called clonal type per kilobase of reads. This is not all the reads in that RNA-seq, but all the reads that are mapped to the T cell specific genes, get like a TCR region of the genome. And so this gives us kind of a total T cell in the tumor and ask what is the number of different TCR clones in that tumor relative to all the T cells, which kind of give us an idea of the TCR diversity. And when we look at all the samples across TCGA, each little dot is one tumor. The y-axis measures how diverse the T cells are. Do you see many different clones? Are they kind of even? And the x-axis here, each column is one cancer type. And so one thing that we noticed is the tumors on the left, these usually are tumors that happen in pediatric tumors or face inside that have low mutation burden. Whereas the tumors on the right side, either they have high mutation burden, for example, melanoma. This is from radiation from the sun or smoking. People who smoke have a lot of mutations in their air wave. And mesothelioma is from asbestos, carcinogen. And colon cancer has microbiome. There are other antigens in there, and also cervical cancer, head and neck, and liver cancer very often have viral infections here. So in general, you can see here that cancer types that have more mutations in the tumor, in their tumors, the T cells are also more diverse because they need different T cells to recognize the different mutations or antigens in the tumor. So we can actually use the trust called TCR and TCR diversity to evaluate tumor immune response and overall compare cancer versus normal and see the differences. We can also use the trust algorithm to get B cell receptor repertoires also from the tumor RNA-seq. And so this is similar to TCR. So you can see here, this is the B cell receptor gene segment with a whole bunch of V, a bunch of D, and a bunch of J. And from VDJ recombination, 1V, 1D, and 1J will be combined together, and the closest one will make a productive B cell receptor. And this thing, when it's expressed on the cell surface, it's called a B cell receptor. But it can also be released from the cell surface, then it will be an antibody. And so B cells produce antibodies for a human. And so normally in our circulating B cells, there is also a selection process in the bone marrow before the B cells are released. Most of the B cells that have high affinity to self proteins are being eliminated. And so most of the B cells circulating are ready to detect outside pathogens, viral infection, bacteria, and parasites, and things like that. And so the inactivated B cells have the receptors on the cell surface, but upon the antigen encounter, the B cell will get activated and make clones for itself, make multiple copies of it. And in the process, a number of things will happen. The first is that the antibody sequence during the clone expansion process, when the B cell make it copies of itself, it will create additional small mutations on the antibody, this VDJ sequence a little bit. This is so that it can evolve an antibody gradually to have better and better affinity, binding affinity against the antigen of interest. So initially, you might have a naive B cell which just binds the antigen a little bit, but as it clones itself, it can have mutation. And if this mutation makes the antibody bind worse, then that B cell will not receive enough signal to further expand. But if the mutation makes the B cell bind to the antigen even better, there will be stronger signals for B cell activation. Then it will clone itself and then create additional mutations. So there's like a clonal selection process. The B cells that can make an antibody with better affinity to the antigen will get a chance to expand even more. And so we can see this gradual clonal expansion event. In addition, the constant region, you can see here, initially there is IgM, which is usually for naive B cells, but the constant regions can also change. And when the antibody is released out to the outside, the constant regions can be different. This is similar to when you call 911, there could be an ambulance or firefighter or policeman. So it depends on the different constant regions. When the antibody recognizes a particular tumor antigen, it will bring different signals into the onto the antigen location. And so this is how B cell evolve and activate in the tumor. Trust can call the B cell receptor sequences also from the tumor RNA-seq. And so one thing we do notice is, for example, this is in one tumor, in TCGA, we can see this kind of clonal expansion happening in the tumor. So there are different BCRs that were called, and most of the positions are quite similar, except for these occasional positions. And based on the mutation, we can even figure out the clonal expansion tree, which one was the original and which one, how does the antibody evolve gradually. And the fact that we see these highly similar ones is not random. It's because of the gradual selection. And the fact we see these mutations is an indication that B cells in the tumor is recognizing the tumor antigen and having clonal expansion to gradually evolve an antibody with better and better specificity against the tumor antigen. And we also mentioned that in the antibody, in the beginning, this is kind of the head of the antibody, which recognizes the tumor antigen, that's the VDJ. But there is also the constant regions, which is kind of like the tail of the antibody, which can recruit different help immune signaling. And so as the antibody matures, it will also shift. When the B cell is a naive B cell, it's an IgM constant region. But as the antibody kind of become clonal expanded, most of the memory B cells are IgG. And so what happens is that in order for the antibody to make an IgG, there's additional DNA cutting on the genome. So basically, if this region of the DNA is being cut out, then the IgG will be the constant region. But if all these beginning ones are being cut out on the DNA, then this one will be the constant region for the antibody. And if all the beginning part is cut out, eventually it will be the later one, the IgA antibody. So you can almost think that this antibody, this is kind of like a young antibody. It still has the potential by deletion to create the later antibodies. But if you have an IgA antibody, it's kind of like the late stage old antibody, because once the DNA for the early regions are being deleted, you can't get them back again. And so because in the nowadays, most people do RNA-seq with paradigm sequencing, very often we can assemble the VDJ sequence from one end of the read, and we can actually figure out what the constant region is from the other read end of the read. And so normally in the tumors, we see these type of class switch recombination. You would expect, for example, the IgA is known to be a kind of a suppressive antibody. For example, all of our gut lining and airways are covered with antibodies. You can imagine this is kind of a sticky line, a slimy region with antibodies just to protect the gut lining. Because if you eat food from outside, there is no point for your immune system to attack the food as foreign, and so the antibody kind of creates a lining to say, okay, you know, whatever is what you eat or what you breathe, no worry about it. And so the outside T cells don't really go in there to attack. But interestingly, we also see a lot of tumors that have this type of antibody class switch. So each tumor, you can see there are some yellow, these are IgM naive antibodies. And we see a lot of pink ones in here, pink and purple ones, which is IgG. IgG are the ones that are clonally expanded and potentially protecting the patient and helping to eliminate the tumor by recognizing the tumor antigen. But then there are also tumors that have a high IgA. And you can see the ones that have really high IgA, which is, you know, green antibodies in here, are colon cancer and rectal cancer. This is similar to, let me see, where is gastric cancer? STAD, yeah, so the gastric cancer also have, you know, this light green is also IgA1. And so you can see they have, you know, a lot of IgA antibodies, because as you know, eat food, there are a lot of foreign things there, then you want to kind of use the antibody to line up the GI system. But you can see here, many other cancers which are not really facing the outside also have these type of IgA antibodies, which is kind of interesting. And so in these non-mucosal tissues, and so we ask, what is the, you know, antibodies really doing in here? You can see here, people mostly believe that IgG 1, 2, 3 are good. And towards the later half, which when the antibody kind of is growing older, especially IgA1 and IgA2, they actually have negative signals. So these, the IgG 1, 2, 3 are mostly kind of activating the immune system, whereas IgG4, IgA1 and 2 are suppressing the immune system a little bit, or at least they are not necessarily killing anymore. And so this kind of helps us understand that, for example, when you have PD-1 and PD-L1 antibodies, both are developed by therapeutic companies to block the PD-1, PD-L1 interaction, but their constant regions are different. For example, for PD-1, because PD-1 is highly expressed on the T cells, you want to have an antibody that blocks the interaction, but you do not want the antibody to bring in help to kill your target T cells. Therefore, the current immunotherapy for PD-1 is using IgG4 as the constant region. So you want to kind of at least not doing the killing, but you don't want to make it too suppressive. You don't want to suppress the T cells. But in here, it's just kind of having a blocking function. But when the antibody against PD-L1 is developed, because the PD-L1 is kind of expressed on the cancer cells to block the T cells. And so when you develop an antibody, you not only want to have blocking, you also want to kind of bring in the immune activation to kill the cancer cells. And so there, the anti-PD-L1 antibody use IgG1, which has kind of more immune activation and killing effect. So the antibody, if it binds to the PD-L1, will bring killing to the target cell. And so understanding these different constant regions, you can see that different constant regions actually does different things. And so this is one recent study that's looking at the the survival of the IgG versus IgA. This is in melanoma, another solid tumor. And they can also see that in the tumors, if there are more IgG antibody compared to all the antibodies, the patient usually have better survival. But if they have more IgA antibodies, then the patient usually have a poor survival. This is similar to our previous observation that IgG1 is kind of a good antibody, whereas IgA antibody is kind of the old suppressive antibodies. We also observe this in leukemia patients. This is with children and infants where they don't have enough IgA2 yet. Infants usually have very young antibodies in their blood. And if they even have more IgA1 type of antibody, you can see these infants or children, they will die more. Whereas in the adults, the antibodies are growing kind of older and older. You see more IgA2 antibodies. And indeed, when the adult leukemia patients have more IgA2 antibodies, they also have worse survival. And so in the tumors, cancer cells sometimes can secrete cytokines to force the antibody from a killing type to a suppressive type. And one important cytokine is TGF-beta. You can see here in the tumors, if the tumor has more TGF-beta, the antibody will shift more towards the IgA type. But if you have less, the tumor has more IgG antibodies, which means that antibodies in the tumors are recognizing the tumor antigen. It's trying to do the killing, but the cancer cells can secrete the cytokines and force the B cells to shift its antibody from a young, good IgG antibody to an old, bad IgA antibody, and then can eventually even potentially help the tumor growth. But it also indicates that if we give back the patient those IgG antibodies, it might actually help cure those patients. So that's on the tumor TCR, BCR. The next question we want to ask is, what controls T-cell mediated killing of the cancer cells? We mentioned that the T cells in the tumor are using the T-cell receptor to recognize antigens that are presented on the cell surface. If the cancer cell present a foreign antigen which can be recognized or can have very strong binding to a particular T-cell receptor, that T-cell will be activated. And activated T cells have ways to kill the cancer cells. There are many molecules on the cell surface that can, that are called immune checkpoints. They either stimulate T cell activation or suppress or inhibit T cell activation. And over the years, people found many such immune checkpoints. And so you can see here the ones that are labeled in blue, like the stop sign, these are the inhibitory genes, which will make the T cell kind of more suppressed. And so the T cell won't get a chance to be activated. This is just to make sure that you don't want to have continued T cell activation and keep attacking and attacking. But there are also molecules which can activate T cells. And so these have the green arrows in here. And so you can see, and a lot of them also have interaction partners on either dendritic cell or on macrophage or on cancer cells in general. So this is not just dendritic cell. It could mean, what other cells in the tumor. When this pair interacted is often when this gene will be activated, and then they will have either their suppressive function or their activating function. And so over the years, people have been trying to develop antibodies that, for example, block this action in order to block those co-inhibitory or inhibitory molecules. And some people also develop antibodies against these molecules as agonists to help stimulate them. and so that the T cells can be activated. But one way to understand the T cell activation and also identify these molecules is to study them in vitro, meaning outside the body in cell lines or cell cultures, in vivo, meaning in mouse, and also potentially in human tumors. And this can also be done either in bulk RNA-seq or single cell RNA-seq in recent years. And so when the T cells are activated, it can use 3 different pathways to effect a cancer cell killing. The first thing, these are called cytolytic T lymphocytes, or basically killing T cells. And so there are 3 possible ways. The first is once the T cell receptor recognizes the target cell as being foreign or infected or bad, the T cells get activated. It will secrete some cytokines such as tumor necrosis factor or interferon gamma. So these are kind of toxic factors. You can imagine they are like bombs. And so they secrete these cytokines. The cytokines do a number of things. For example, for interferon, in the cancer cells that are receptors such as TNF receptor or interferon receptor, they receive the interferon signal, and a lot of those will induce the cell to die, because it's kind of like, you can imagine this as a bomb. You secrete the cytokine, and then eventually the cell will die. But then if there's low level of that, 2 more things will happen. One is that the cancer cell will upregulate the CD95, which you will see here later. The other is that it will upregulate the cancer cell to overexpress the MHC class 1 molecule. Remember, MHC class 1 is presenting things, antigen onto the cell surface. And so if the T cell detected something being foreign, it will secrete the cytokine, which forces the cancer cell to bring more things onto the cell surface to be examined. And so it's like, if a guard sees something suspicious, it will just keep probing, right? Where do you live? Where do you work? Who's your parents? Or, like they just keep asking more questions in some sense. And so the interferon is forcing the cancer cells to present even more antigens onto the cell surface for examination. And so this is the effect of interferon on the cancer cells. It will make the cancer cell die. It will force the cancer cell to overexpress CD95 and also the antigen presentation. In addition, interferon is a signal that allows the T cell, it will stimulate the T cell to make clones for itself. So it makes more copy of the same guard. And third, interferon create a gradient. So things that are closer to the activated T cell will have higher concentration of interferon, and further away will have lower concentration of interferon. And other T cells will be like a shark smelling the blood. They're like, okay, something went wrong over there. And so other T cells will follow the interferon gradient to come into the tumor and help the initial activated T cells to examine the cancer cells for additional mutations and additional activation. And so that's the first way. The second way for the T cells to kill the cancer cells is through this Fas ligand. So it creates a kind of like a signal again on the cell surface. This instead of interferon, which is released outside without direct contact, Fas ligand is a ligand on the T cell surface, which can interact with CD95, which is a receptor on the cell surface. And once these two interacted, this will also activate the downstream caspase pathway to have the target cell die. And so remember, when the T cells secrete interferon, it's going to activate the CD95, which is the receptor. And when the T cells express the ligand to interact with this receptor, again, the cancer cell will be induced to die. And the third one, also this is through contact. So you can see when the TCR is interacting with the foreign antigen, there is a process where on the interaction surface between the T cells and the cancer cells, the T cell has something called porphyrin, which is a protein that can punch a hole on the cancer cell surface, and then secrete granzyme, which is another type of kind of a toxin into the cancer cells through those holes, and those are packaged into little granules of poison and sent to the target cell. And so when granzyme are released in the target cell, the cancer cell will die again. And so this is also through contact of the two cells and the granzyme punching the hole right at the two cell interaction surface. And so these are the three different modes of how an activated CD8 T cell can, also CD4 T cell can be activated for this type of killing. And so in order to really look at what the T cells are doing, people now are looking at single cell RNA-seq. For example, this is one very early study in 2017 where the scientists used the tumor RNA-seq compared to the blood, and then they collected all the T cells, CD4 and the CD8 T cells, and did expression RNA-seq analysis. And through trajectory analysis that you learned from the course earlier in module 3, they can look at the cell trajectory to see what happened to CD4 T cells or CD8 T cells. And what they found was that, for example, in CD4, initially, before the T cell receptors are activated, you have naive cells. And then once the T cells recognize the foreign antigen, it will get activated. It will become a effector T cell, which kind of starts to overexpress granzyme and also porphyrin. And cytotoxic, meaning that they are starting to do the killing. At this stage, there's also proliferation of the T cells, but once the T cell have tried enough killing and proliferated enough, eventually it will become exhausted and then die. This is on CD4. CD8 is the same. Initially, it's a naive T cell, and then it gets become an effector. You can start to see interferon expression, granzyme activation. So you see the effector T cell, memory T cell, and also cytotoxicity happening in the T cell. But then eventually the cell will be exhausted and die. And so from the single cell RNA-seq, we can actually look at these gene expression differences and see the trajectory of T cell activation and exhaustion. And so one more detailed study looking at, in this case, immunotherapy patients, they look at both before, this is only after this is before and after immunotherapy. I remember not all the patients, but some patients have before, some patients have after, and some are responding to immunotherapy and some are not responding to immunotherapy. And they selected all the immune cells for single cell RNA-seq analysis. And then they ask, what are the difference between the CD8 T cells from those responding tumors versus those non-responding tumors. So this is the CD8 T cells from the bad patients versus the good patients. So they assume that CD8 T cells are kind of different between the responders and non-responders. Yeah, so and when they look at the CD8 T cell gene expression difference, they saw the one important difference, which is TCF7. This is in the naive CD8 T cells. And when the T cells are activated, if the patient can retain enough of the TCF7 positive CD8 T cells, then they observed a better response and also overall patient better survival. This suggests that when the T cells are activated, you'll make too many copies of itself and then eventually it will crash and burn. But if you have enough of the original naive cells, the naive cell is like a stem cell-like. It can continue to replenish the microenvironment with additional new young T cells to be activated and proliferating. Then the patient will have kind of a better response to immunotherapy. So when you have fresh supply of armies to kill the cancer cells, then the patient will have better response and better survival. And so you can see here, this is again from our single cell RNA-seq analysis, and just looking at the differential expression, they were able to look at the TCF7 gene expression to be different between the good and bad CD8 T cells. We also mentioned that T cell can become exhausted. And so this is a study published in 2017, just using bulk samples. They didn't really treat the mice with tumor. Actually, I think this is treated with kind of a viral infection for a very long time. And initially, this is day 0 when the antigen is being infected. This is like using infection to activate T cells. And initially the T cell get activated, and then they just continue the infection, and they wait for 5 days, 14 days, or 28 days, 35 days, and 60 days, and then collect the mouse T cells to see the difference in the mouse T cells. And they can see this, from the early to the middle and to the late, especially the 60-day late. And they can see some differences. For example, before the cell is activated, they don't produce interferon. And once you have the infection, the T cell become activated, it will secrete interferon. As we mentioned, it's one way for the T cells to kill the target cell. And so you can see the T cell expressing the interferon. But over time, this particular enhancer, this is doing ATAC-seq to look at epigenetic changes on the cell. The cell's ability to produce interferon could decrease over time, and this is because the cell over time is losing an enhancer in here. This is another gene, which is PDCD1. This is the PD1 gene on the T cells. You can see initially PD1 is slowly expressed. There's very low signal. In the early viral infection, also there's low signal. But then in the late, you can see this region have a new enhancer. There's another, this enhancer decreased, but this enhancer increased. And then you have a overall increase the PD1 expression in the later stage of the T cells. And so using this type of gene expression and RNA-seq, RNA-seq and ATAC-seq, this group was able to generate a signature for what they call early T cell dysfunction or exhaustion markers. And so in these early days, or like this, basically the medium, is like when you just started to see the T cell becoming a little dysfunctional, but towards the end, you see the much more exhausted T cell markers. And what once they have these markers, they evaluated, whether PD1 antibodies can reverse this level of T cell dysfunction. And what they found is that when the cells are still in the early dysfunctional state, PD1 antibodies can actually, or PD-L1 antibodies can reverse that dysfunctional state. But in the fixed, or in this case is epigenetically fixed T cell dysfunctional state, PD1 is no longer able to wake up or to reactivate the T cells. And so you can imagine, if you think about T cells as guards trying to kind of look for the bad guys, this early stage is when the guards are just getting tipsy. And so if they're just tipsy, you can treat them with PD1 and wake them up. But in here, the T cells are like dead drunk. And so you can't really use PD1 to wake them up anymore. And so this is another study published in 2019, looking at what factors are really driving the fixed T cell dysfunction. And so they look at the CD8 T cells, and again, they did ATAC-seq. And they want to see the super enhancer analysis. This is just, you can imagine this is looking at a taxic regulatory potential on all the genes, and they see what are the top genes and what are the later genes that are showing difference. And so they see this transcription factor called TOX. In the naive T cells, the rank is 91. And in the effector, this is the T cell just getting activated, TOX dropped to 365. But then when you have memory T cells, that's when you start to see early exhaustion marker. You see the TOX becoming rank 64. But then in the very kind of fixed dysfunctional state, they see this TOX ranked at 35. And this is actually a potential indication that epigenetically, this gene is becoming more and more important. And turns out this is a transcription factor that they discovered as a very important regulator for T cell exhaustion. And so this is from John Waring's group when they try to overexpress this TOX transcription factor, and they look at the differential genes in the CD8 T cells. They see a lot of other T cell dysfunctional markers. PD-1, this is TIM3, also other markers for T cell exhaustion. And so you can see you can use single cell RNA-seq, you can use epigenetics or ATAC-seq to really look at either tumor or chronic infection to examine how T cells get activated and when they start to see early exhaustion, and also this type of fixed exhaustion status, and whether the cell still maintains some progenitor population to continue to replenish the activated T cells. And these all have implications to immunotherapy response. The next question a lot of immunologists really want to learn is what other cells can be activated or how they can be activated also to kill the cancer cells. Overall, the biggest killer, the most important killer that is very important in cancer immunology is T cells, CD8 and CD4 T cells. They can do the killing. But there are other cells that are also important. Here we mentioned myeloid cell, but you can imagine this also being cancer cells. If they present some antigens, this is kind of myeloid cells presenting the dendritic cells, also myeloid cell. They present the tumor antigen onto the surface, but this can be cancer cells directly. They also present antigens or mutations onto the cell surface. And these antigens, they can be recognized by T cells with T cell receptor, but they can also be recognized by B cells with antibodies, right? So once the antibodies bind to the tumor antigen, a number of things can happen. The first is that there's a process called the complement-dependent cytotoxicity. You can see here, once the antibody recognize the tumor antigen, they will recruit this CDC or the complement system, and then recruit another complex called MAC complex. And this MAC complex will again punch holes onto the cell surface, and the cell will lyse and die. And so this is one way the antibody recruits complement and MAC to punch holes onto the target cell. The second approach is antibody-dependent cellular cytotoxicity, or ADCC. And so this is when the tumor antigen is being recognized by the T cells. If it has the right constant regions, in this case, for example, IgG1 and IgG3, they will be able to interact with the Fc, the receptors, on the natural killer cells, so that they can bring the natural killer cells to the location. And the natural killer cells will kill the cancer cells. And a third is macrophage. This part is quite similar. When you have an antigen being recognized by the antibody, again from the Fc gamma receptor, it can recruit a macrophage. And when the macrophage comes, it doesn't directly kill, it engulfs, it eats up the target cell. And this is especially useful when you have a bacteria that is really foreign, and the macrophage will eat up the bacteria and then chew it up and spit it out and present those onto the cell surface. And so you can see B cells, they don't necessarily, well, directly kill, but through CDC, ADCC, and ADCP, they can recruit other members there to do the killing. And so with natural killer cells, they can they kill with the following mechanism. So normally, all the normal cells have to express MHC class 1 to present antigen onto the cell surface. And T cell receptor makes sure that the presented antigen is not foreign. They have seen this before, and so they don't get activated if it's normal antigen. But natural killer cell also has a receptor to ensure that all the cells need to have MHC presentation, because one way for the cancer cell to evade T cells is to lose the MHC. And so basically, if the cancer cell does not present things onto the cell surface, then the T cell cannot recognize it anymore. And the body has a way to deal with this. Our natural killer cells ensure that all the cells have to present MHC class 1. If they don't, if they don't have ID, that's definitely bad. And so, if the bacteria come in, they don't display MHC class 1, NK cells can get activated very quickly to say, this thing completely do not have ID, let alone having the correct ID information. And so they do the killing. There are also, so you can see here, when the cell is missing the class 1 presentation, NK cell can kill. In addition, sometimes the cancer cell can have signals to when it's under stress. When the cell is under stress, there are ligands that are activating the NK cell. So the MHC class 1 is a suppressor. If this is present, the receptor can recognize this and NK cell does not get activated. And so if you don't have the suppressor, the NK cell will kill. Additionally, if the cancer cell is under stress, there are upregulated ligands, which will activate NK cell even more, then the NK cell will still do the killing. And so in this particular case, the cancer cell may not lose the MHC class 1, but if it presents an antigen and through ADCC can recruit the NK cell to the surface, the NK cell will be there and then see whether this cancer cell express some stress ligand on the cell surface. And if the stress ligand is activated, then the NK cell will again kill. Basically, the Fc gamma receptor already recognized the cell as expressing something foreign, and the cell is under stress, then the NK cell will know that this is a cancer or infected cell that presents foreign antigens, then you will kill it. And so a lot of these T cell activation and cancer cell relationship, you can also see that in the tumor infiltration and also the NK cell expression data from single cell RNA-seq analysis. And we mentioned another player, which is macrophage. Macrophage can engulf the cancer cell, but there are two different phenotypes of macrophage. In our blood that's circulating, we have very little macrophage. It's mostly called monocyte, which is a progenitor of macrophage. But once the monocyte infiltrates into normal tissues or into the tumor, it kind of differentiates into either M1 macrophage or M2 macrophage. And these two also have very, very different phenotypes. The M1 phenotype is more immunostimulation, inflammation, and anti-tumor activity. It's mostly to, it's similar response to recognizing a bacteria and trying to eat it up. And so the M1 macrophage is good. Once it recognizes the tumor, it will try to kill it. So it's more a similar response to a bacterial infection. Whereas the M2 macrophage is kind of more for tissue repair, remodeling, and it's more immune suppression. This actually will hold the tumor growth. And this is a wound healing phenotype. And so normally when you have a cut, there will be some bacteria and germs coming in here. And there's like a lot of macrophage that goes there and kind of form like a cell band-aid. And so this allows the cell to kind of wound to heal, because as the wound is healing, there will be some things from outside. And if your immune system keeps on attacking and having too much inflammation, that wound will never heal. And so macrophage kind of, once the macrophage in the M2 is covering that region, it kind of insulates the outside from the inside, because outside there will always be some bacteria and bad things. With macrophage insulating there as a bandage, there's no point to get alarmed, whereas the inside will be kind of self. And so the cancer cell have also learned this trick, and you can pretend to be a wound and force they secrete the cytokines to force the macrophage to differentiate from an M1 to an M2 type. And once the M2 type is in the tumor, the macrophage is no longer being activated. Those M2 macrophage are mostly suppressing the other immune system and not allowing the T cell to get activated. And so in each patient tumor, you can always see macrophage infiltration, some M1 and some M2. And very often we see that patient with more M1 macrophage phenotype usually have better survival and responding to immunotherapy. But if they have more M2 macrophage, it's usually pretty bad because they will suppress the T cells. And so you can see this is just one example of another study looking at single cell. You can do either single cell RNA-seq or CyTOF. They are looking at this is lung cancer, and they use specific antibodies even to select to kind of enrich for specific cell types. And then they do the single cell profiling. And then when they look at the changes in normal lung tissues versus lung cancer cells, and they can see the microenvironment really changing. For example, they see that there are clonal expanded PD-1 positive T cells, meaning that they can see the T cells becoming a little bit exhausted. They see an increase of CTLA-4 high T regulatory T cells. Tregs is also bad. It kind of suppressed T cell activation. And they see the PPAR gamma high macrophage. This is kind of a macrophage go more towards the M2 phenotype. They see a decrease in NK cells, and they also see two different dendritic cell populations. So the CD141 dendritic cell is kind of a good dendritic cell. It's better at presentation to the T cells for T cells to recognize tumor antigen. But unfortunately, those T cells are decreasing, whereas the worst type of dendritic cells, CDC2 with high CD1C level, they are increasing. And so you can see the single cell analysis can really see these differences, suggesting that if there are drugs that can reverse this trend, increase NK, move the macrophage to M1, increase antigen presentation, and suppress Tregs, that can help with immunotherapy response. The next question is how to predict immunotherapy response. So with the current cancer immunotherapy results is really exciting, because compared to previous drugs, which work only for a very short time and the patient develop resistance, and also there are drugs that patient has to keep taking forever. Immunotherapy is kind of interesting in that it stimulates your immune system. And once the immune system is activated, it will do the job. And some patients can be cured, after say 10 shots. they are totally cured and they don't have to take the drug anymore, which is very exciting. Unfortunately, this is just only a minority. By this time, a lot of clinical trials are being conducted in different cancer types to predict immunotherapy or to evaluate immunotherapy efficacy. And so far, the ones that worked very well are melanoma, lung cancer, which remember have a lot of mutations. They look very foreign to the T cells compared to normal cells. But in many different cancer types, even though the drug has been approved for usage, only a minority of patients really responded. For example, as a single therapy for breast cancer, it only works in the ER-negative breast cancer or triple negative breast cancer, but only works for less than 20%. In non-small cell lung cancer, it's also about 20, 30% as a monotherapy. With melanoma, it's probably 30, 40%. And so the big question is how to really predict response to immunotherapy. Which patient will really respond to immunotherapy? Over the years, there are many, many different biomarkers that are proposed and also being evaluated in different immunotherapy trials. And so there are some kind of well-known ones. The first is T cell level, which means whether the tumor, the T cells are infiltrating the tumor. You can imagine the guard example before. If T cells are like guards, you want to make sure there are many guards already in the tumor. Maybe they are only tipsy, so you can wake it up. So if in a tumor you don't see any guards, whether or not they're drunk or not, then it's not very good. The second one is T cell diversity. As we mentioned, in order for the T cells to recognize the mutations, if the tumor has, if the T cell have different TCRs, many different TCR, just by chance, one of those TCR recognizing a tumor antigen would be a little higher. And so young patients with diverse T cells are more likely to respond better to immunotherapy. But in general, whether it's in the blood or in the tumor, people see that if you have higher T cell diversity, it's probably, at least in the blood, is a good thing. In the tumor, because it's kind of difficult, sometimes the correct T cell is already clonally expanded. And so that clonal expansion might hide some information about the overall diversity. So the result hasn't been as consistent. But overall in the blood, you want the T cells to be quite diverse. And the next one is PD-L1 levels. This one is kind of puzzling. PD-L1 is, cancer cells or sometimes macrophage or B cells also express PD-L1 on the cell surface. Is the cancer cell checkpoint to inhibit T cell from activating, to say, hey, buddy, let's be friends, no point to get alarmed. And so the PD-L1 interacts with the PD-1 to suppress T cell activation. And so most people think that if you have higher PD-L1, the patient should do worse because it means that the T cells are being suppressed. However, because we want to make sure that the cancer cells antigens have already been recognized by the T cells, and the T cells initially probably try to attack, that's when the cancer cells will upregulate PD-L1. So PD-L1 is usually activated when the T cell has just started killing or secreting the interferon or secreting the granzyme and porphyrin to the cancer cell. That's when PD-L1 get activated. And so if we see higher level of PD-L1 expression on the cancer cells, this is an indication that the guards were trying at least to kill. And so you can imagine this is kind of some signs that the T cell have been activated before. And so that's kind of a very, very important biomarker. In fact, the Merck, they are the most successful in the current PD-1 antibody therapy market. and they very often use PD-L1 on the cancer cell surface level. They usually ask, does 30% of the cancer cell have PD-L1 or 50% of the cancer cell have PD-L1 protein level on the cell surface? They use that as a biomarker for predicting immunotherapy response. And then another one that is really important is also tumor mutation load. As we mentioned, if the tumor looks really foreign, it has a lot of different mutations. Some T cells will likely to see this as foreign. They are more likely to recognize this tumor and then get activated, right? So yeah, this is one study looking at immunotherapy in melanoma. And so the patient have DNA damage repair defects. So when they have MMR defects, their tumor has a lot of mutations, not just in melanoma, in say colon cancer. If the tumor has DNA damage repair defects, they will accumulate a lot of mutations. And interestingly, those tumors with the a lot of mutations, their tumor will shrink. Whereas those that have correct DNA damage repair mechanism, their tumor most likely don't respond to immunotherapy. And so you can see here, many different biomarkers are being predicted. So overall, in terms of tumor immune evasion, meaning that how does the tumor initially develop, and also immunotherapy resistance, meaning that they don't respond to, for example, the current PD-1, PD-L1 antibodies, there are a number of these different mechanisms, right? So this is a review in 2019 where they mentioned a few things. So let's first look at this first case. They are called immune hot tumors, meaning that there are T cells in the tumor. It's trying to do the killing, but then those T cells might be suppressed with exhaustion markers such as PD-L1 or TIM3, LAG3. And so then the T cells are there, they are just drunk. So immunotherapy hopefully will bring the tipsy T cells back into action. But then there are also cases when the T cells cannot really infiltrate the tumor. And so over time, people have discovered a number of factors that prevent T cell from activating. This includes myeloid derived suppressor cells, tumor-associated fibroblasts, high level of T regulatory cells, and also the M2 type of macrophage. Remember, the M2 is a wound healing. It's like, T cells don't come here. There's a wound, let it heal. Don't need to attack outside. And so, so with these suppressive cell types, then the cancer cell really cannot infiltrate. And also, a lot of these immune cells are going into the tumor through blood vessel. And so there has been, I would say, conflicting results about whether blood vessel is good for immunotherapy or bad for immunotherapy, but at least different studies have evaluated, and they do see some differences. Some say, having blood vessel is good, at least you bring the T cells into place, but other ones also say that it's bringing other, bad cells into the tumor microenvironment. There are, and then there are also factors that make the tumors cold or the T cells don't get activated, which is in this case dendritic cell, whether the dendritic cell is presenting the antigens well enough to the T cells or not. So one is the dendritic cell activity. The other is whether there are MHC presentation defects. So antigen presentation, if they are defective, for example, the cancer cell may lose MHC1, then their mutations never get presented onto the cell surface. That's preventing the T cells from recognizing the antigen. And the final case is lack of tumor antigens. If this tumor have low mutation burden, very low mutation, then they don't usually benefit from immunotherapy. And so you can see all of these different mechanisms. So far, each study only evaluates one or two. It's hard to really develop good predictor that works on all of them together. So within our lab, we also have a computational algorithm. We call it TIDE, which stands for tumor immune dysfunction and exclusion. We evaluate both T cell dysfunction and T cell exclusion. And so the algorithm uses the pretreatment tumor bulk RNA-seq biopsies to make the prediction. And based on the bulk RNA-seq, we can estimate the level of CD8 T cells that are infiltrating the tumor. We can also evaluate whether those CD8 T cells are activated by monitoring the granzyme and porphyrin levels in the tumor. And so from the tumor expression, we can tell whether this is an immune hot tumor or an immune cold tumor. So immune hot tumor are those that have enough T cell infiltrating, and you see the granzyme porphyrin there. And what we found was that if those T cells are in a fixed dysfunctional state, meaning the guard is too drunk, then immunotherapy would not be able to wake it up. But if the T cells are, it's kind of immune hot, the T cells are just a little bit tipsy, maybe immunotherapy can bring it back again. Whereas for the immune cold tumors, there are a number of factors, whether the tumor have M2 macrophage, cancer-associated fibroblasts, or other myeloid suppressor cells, or maybe Tregs, or low antigen presentation, maybe the tumor is not really hot. And those will also not respond to immunotherapy because the current immunotherapy is trying to revive the T cells, but if the T cells are not even infiltrating the tumor or things preventing the T cells from infiltrating the tumor, then the T cells can't be activated and do the killing. And so we have this website and we have been accumulating a lot more immunotherapy clinical trial data, and also we are implementing additional reported immunotherapy response biomarkers. And so we have this paper that has a number of functions. The first is a biomarker evaluation feature. So if you come up with your own immunotherapy biomarker, you say, okay, these genes are important and these are bad, these are good, and you can use the weights to give it different weights or different signs. And then our algorithm will use all the published immunotherapy trials to try to see how well your biomarker is predicting the response compared to all the other ones, T cell infiltration, mutation load, PD-L1 level, CD8 infiltration, and so on. And another is a response prediction algorithm. And so if you have this is for oncologists. If you have many different patients, each with a pretreatment tumor RNA-seq, you can enter this here and use a number of these different biomarkers to try to make predictions. And so if every biomarker says that this patient is not going to respond, well, then they probably shouldn't be treated with immunotherapy. And if, yeah, so this is actually from TIDE, and you can see here what TIDE predicted to be responders indeed were responders, and the TIDE predicted non-responders were non-responders, although we do make some mistakes in here. And so if the oncologists can enter the trials, we can help with prediction as well. And so in the recent DREAM challenge of predicting immunotherapy response, we actually did pretty well in that challenge or competition. And the third one is a regulator prioritization function. This is for cancer biologists. If you do a genomic experiment, you might see many genes as upregulated, say a single cell RNA-seq, right? You see many genes that are interesting. Which one do you go? Because usually you have done expression analysis. You see hundreds of genes as being differentially expressed. Can you predict which one to be important? Because most of the single cell data are generated on a very limited system, whether it's a mouse tumor or very limited patients, we can use many other clinical trial samples or non-clinical trial, but patient tumor with survival information, and also CRISPR screens, which we'll teach in the next lecture, to help figure out which one is really important. And so to demonstrate this, recently we were in a study. We know that the glycosylation regulators are potentially important for cancer cell to evade the immune system. So if the cancer cell have a lot of sugar on the tumor surface, this can kind of suppress the T cells. And so we ask what kind of these glycosylation enzymes are the most important. And we put this into the TIDE website, and TIDE, using all the other data, help us prioritize this top gene as MAN2A1. And it turns out this gene has a drug. And we were able to use all the clinical trial to see, in which trial does MAN2A1 inhibitor synergize with current immunotherapy. And it turns out it is with PD-L1. And so we were able to experimentally validate that indeed, when you combine the MAN2A1 inhibition, this small molecule drug with anti-PD-L1 antibody, it can make the tumor shrink faster. So this is kind of using all the immunotherapy data and existing biomarkers to predict the future patient response, and also potentially predict future better ways to improve immunotherapy response. The last question we want to answer is how to improve immunotherapy response. And so we mentioned that most of the immunotherapy trials only work well in 20–30% of the patients, right? And it's very important to improve immunotherapy. And the key is combination therapy. For example, previously people have tried just to use anti-PD-1 or just anti-CTLA-4, but this is an example of a trial published in New England Journal of Medicine. They see that even though the single drug anti-CTLA-4 in melanoma, the response rate is only 11%. For PD-1, I think it's a little higher, probably 30%. But when you combine PD-1 with CTLA-4 together, two drugs, the response rate is 61%, much, much better. Because they work, the CTLA-4 and PD-1 are still kind of complementary mechanisms. Each make the tumor environment a little better and more activated for the T cells. This is another example of anti-PD-1 combination with chemotherapy. In the red, it's just chemotherapy alone in non-small cell lung cancer. This is survival for the progression-free survival. On the right side is the overall survival. So you can see when you only use chemotherapy, this is kind of the survival curve. But if you combine anti-PD-1 with chemotherapy, the response is much, much better. And these, by the way, for this particular trial, they required a patient to have over 50% of the cancer cells expressing PD-L1. And those are the patients that are allowed to come into the trial. And then you can see those patients really benefited from an anti-PD-1 antibody in combination with chemotherapy. And this result is even better. It's not only better than chemotherapy, later on, there are also trials to show that this is also better than anti-PD-1. Because very often the cancer cell, when it's treated with chemotherapy, will increase antigen presentation, which will then help the T cell to recognize the tumor antigen. And then with the PD-1, the T cell won't get dysfunctional. So this is a review paper from Pam Sharma, where he see that, in chemotherapy, you kind of prolong the life a little bit, but eventually patient will become resistant to the drug. In targeted therapy, you see a very good response, but also only prolong the patient. Initially, remember the BRAF inhibitor, it's like a magic cure, but only for a little bit. Eventually the patient will develop resistance, and most of them will die from drug resistance. The nice thing about immunotherapy, even though only a minority of those patients responded, the responders, especially those that responded to CTLA-4, it seems like they can get completely cured, totally tumor-free for very, very long time. This has also been observed for some of the PD-1 patients. And the hope is that if we combine different immunotherapy, combine chemotherapy with immunotherapy, combine targeted therapy with immunotherapy, we might be able to reach a curve like this. So you can see this is the beginning of that. But maybe we can really combine them in a better way. That's when we really see precision medicine based on the tumor profile. We tell them, okay, you should use this targeted, this chemo, this radiation, this immunotherapy. And maybe we will get to this point. And so in order to really identify those combination, identify also novel targets to develop new drugs, or identify existing drugs that can be combined with immunotherapy, we will need additional tumor profiles and also CRISPR screens, which we will talk about in the next lecture.